Chapter 12, Section 1, DNA. How do genes work? What are they made out of? And how do they determine the characteristics of organisms? Are genes single molecules, or are they longer structures made up of many molecules? In the middle of the 1900s, questions like these were on the minds of biologists everywhere. To truly understand genetics, biologists first had to discover the chemical nature of the gene. If the structure that carries the genetic information could be identified, it might be possible to understand how genes control the inherited characteristics of living things. Griffith and transformation. Like many stories in science, the discovery of the molecular nature of the gene began with an investigator who was actually looking for something else. In 1928, British scientist Frederick Griffith was trying to figure out how bacteria make people sick. More specifically, Griffith wanted to learn how certain types of bacteria produce a serious lung disease known as pneumonia. Griffith had isolated two slightly different strains or types of pneumonia bacteria from mice. Both strains grew very well in culture plates in his lab, but only one of the strains caused pneumonia. The disease-causing strain of bacteria grew into smooth colonies on culture plates, whereas the harmless strain produced colonies with rough edges. The differences in appearance made the two strains easy to distinguish. Griffith's experiments. When Griffith injected mice with the disease-causing strain of bacteria, the mice developed pneumonia and died. When mice were injected with the harmless strain, they didn't get sick at all. Griffith wondered if the disease-causing bacteria might produce a poison. To find out, he took a culture of these cells, heated the bacteria to kill them, and injected the heat-killed bacteria into the mice. The mice survived, suggesting that the cause of pneumonia was not a chemical poison released by the disease-causing bacteria. Griffith's experiments are shown in Figure 12-2. Transformation. Griffith's next experiment produced an amazing result. He mixed his heat-killed disease-causing bacteria with live, harmless ones and injected the mixture into mice. By themselves, neither should have made the mice sick, but to Griffith's amazement, the mice developed pneumonia and many died. When he examined the lungs of the mice, he found them filled not with the harmless bacteria, but with the disease-causing bacteria. Somehow, the heat-killed bacteria had passed their disease-causing ability to the harmless strain. Griffith called this transformation because one strain of bacteria, the harmless strain, had apparently been changed permanently into another, the disease-causing strain. Griffith hypothesized that when the live, harmless bacteria and the heat-killed bacteria were mixed, some factor was transferred from the heat-killed cells into the live cells. That factor, he hypothesized, must contain information that can change harmless bacteria into disease-causing ones. Furthermore, since the ability to cause disease was inherited by the transformed bacteria's offspring, the transforming factor might be a gene.